Today in this video about cellular respiration, we will see the beginning of the reactions that will allow cells to harvest energy from carbohydrates. In the cellular respiration part 2, we will see how other fuel molecules can do so as well. So, first of all, the cellular respiration process begins with a first phase called glycolysis, and this is a phase that doesn't need to have oxygen present. This is a phase that can occur as easily in presence or in absence of oxygen. Then the two main outcomes of the glycolytic process are the pyruvate and ATP. Pyruvate is a 3-carbon molecule that will be taken into another pathway which is called the citric acid cycle. Before it enters the citric acid cycle, it will need to be converted into acetyl coenzyme A. We will see that more in details. And the outcomes of the citric acid cycles are ATP molecules and electron carriers. Then electron carriers, which are the real carrier of energy, will be themselves transported through an electron transport chain, and the final outcomes of the process will be water and ATP molecules. So let's first see the first stage of cellular respiration, which is glycolysis. The glycolytic process, which is the equivalent of glycolysis, is basically the splitting of a 6-carbon sugar into 2 3-carbon molecules, and this occurs, as I said previously, in absence of oxygen. So whether oxygen is there or not will not make a difference. This will occur in the cytosol and won't need any oxygen molecule in the process. So there are two phases in the glycolytic process. So the glucose molecule, which is the most frequently entering sugar in glycolysis, will first need to invest 2 ATP molecules. So basically 2 ATP molecules will be consumed by the cell in order to produce 2 3-carbon sugars. And these 2 3-carbon sugars will be used in the next phase that will be called the payoff phase and will give up 4 ATP molecules plus NADH, which is one of the electron carriers. So the last outcome will be the pyruvate molecule. So you see that you have invested 2 molecules of ATP, you gain 4 ATP molecules, so it means that the net result is 2 ATP molecules for the glycolysis. Now let's have a look on glycolysis, a closer look on glycolysis. So I know that the slide may look a bit busy, but we will take things one by one. So if we look at the first phase, the preparatory or investment phase where we consume ATP, you see that the entering molecule in the pathway is glucose. Glucose will be phosphorylated, so it will receive one phosphate. The phosphate will come from ATP, adenosine triphosphates, and ATP will become ADP, so diphosphate, while one phosphate will be attached to the glucose molecule using the energy provided by the release of the phosphate from ATP. And it will be attached on the carbon 6 of the glucose, so it will be called a glucose 6-phosphate. It doesn't mean that it has 6 phosphates, but it means that the phosphate group is added on the carbon 6 of the glucose molecule. This glucose 6-phosphate will be converted into fructose 6-phosphate. It is a reaction that doesn't consume energy. Then fructose 6-phosphate will also receive another phosphate, again from an ATP molecule that will release one phosphate as well as energy, enough energy to attach the phosphate on the fructose 6-phosphate. It will therefore be called fructose 1,6-bisphosphate because the second phosphate is added on the carbon 1. So you will basically have a fructose molecule that has a phosphate on its first and on its last carbon. This fructose molecule will then enter in a second phase, that is a cleavage phase, the cleavage into two molecules. So the fructose 1,6 bisphosphate will become the glyceraldehyde 3-phosphate and the dihydroxyacetone phosphate. So two 3-carbon molecules with each one having one phosphate. The dihydroxyacetone phosphate will be converted into the glyceraldehyde 3-phosphate, so they will become the same molecule. It is just that you have two copies of each of the molecule. So now everything that we will mention will be doubled because we have two of these molecules. So we enter now in the phase 3, or the payoff phase, the one that will produce ATP. So the glyceraldehyde 3-phosphate, again, you have two of them coming from the cleavage of one fructose molecule, will be converted in the 1,3-bisphosphoglycerate, because one phosphate coming from the cytosol will be added on the molecule. There is no need for ATP in this situation because the reaction will spontaneously occur and add a phosphate, an inorganic phosphate, on the glyceraldehyde 3-phosphate. This reaction will not produce ATP, but it produces one electron carrier, that one high energy electron carrier, which is the NADH shown here. So you have two NADH produced from this reaction. We'll see later how NADH will be used. Now the 1,3-bisphosphoglycerate will be converted into phosphoglycerate, so it means that it will lose the phosphate that was located on the carbon 1, and losing this phosphate will also release enough energy to put it into an ATP molecule. So you will take an ADP and take the phosphate group of the bisphosphoglycerate, use the energy released by this release, and put it into an ATP molecule. So with this you produce one ATP molecule per reaction, because you have two molecules of 1,3-bisphosphoglycerate, then you will end up with two ATP molecules from this reaction. Then the 3-phosphoglycerate will be converted into the 2-phosphoglycerate, so it means that the phosphate group is just moved from carbon 3 to carbon 2. It will then be converted into the phosphoenol pyruvate, so it is another type of molecule. Again, this reaction doesn't consume energy, doesn't produce energy, but is happening spontaneously. And final stage, the phosphoenol pyruvate, which is a molecule containing high level of energy, will be converted into pyruvate, and because it contains so much energy, the release of the phosphate group from the phosphoenol pyruvate will provide enough energy again to put the phosphate into an ATP molecule. So because you have one ATP molecule produced per reaction and you have two phosphoenol pyruvate, it means that you have two ATP produced from this reaction. So the final product of this reaction is the pyruvate. And the net, the result of the payoff phase is 4 ATP and 2 NADH. Because we consume 2 ATP in the preparatory phase, the net result of this glycolysis process is a 2 ATP yield. So it's a lot of reactions for only 2 ATP molecules gained, and the outcome of this process is the pyruvate molecule. You have 2 pyruvate molecules, and this molecule is a molecule that also contains quite a lot of energy that can be further harvested in the next stages. It also releases 2 NADH molecules that will themselves be able to be converted into energy by a different process that we will see at the end of this chapter. So let's now have a look on the fate of pyruvate. How can we further extract energy from this 3-carbon molecule? this will happen by the citric acid cycle, and this will first occur with the synthesis of acetyl-CoA and then acetyl-CoA entering into the citric acid cycle. Where does this occur? It occurs in the mitochondrion. Mitochondrion is the site of energy production by the citric acid cycle. Note that you may also see in textbooks that this cycle is called the Krebs cycle or the TCA cycle. Note as well that the citric acid cycle itself doesn't produce a lot of ATP molecules, but it produces a lot of electron carriers or NADH and other types of molecules that will themselves be able to produce energy by another process called oxidative phosphorylation, but it's not directly the citric acid cycle. Note as well that the mitochondrion has several other functions, namely to regulate cell death and to produce steroid hormones. How is the mitochondrion built, or how is it made? You remember that it is enveloped in two membranes. One outer membrane that is relatively permeant to a lot of molecules, including big and small molecules. The inner membrane is a bit more selective, but still allows the pyruvate to get into. So you will separate the cytosol from the interior of the mitochondrion into several compartments. You have inside the mitochondrion, so after you've passed the outer and the inner membrane, you will find the matrix of the mitochondrion or the mitochondrial matrix. And between the outer and the inner membrane, you will see the intermembrane space, which is different from both the matrix and the cytosol. Now let's come back to the pyruvate molecule. So we produce the pyruvate molecule in the cytosol after glycolysis. Remember that each glucose molecule will give rise to two pyruvate molecules. If you look closely at the pyruvate molecule, you notice that the end of the molecule contains a group that has a C and two O. So it looks quite like a CO2 group. And what will happen is that when a pyruvate will move through the membrane, it will lose its CO2 group, but instead it will gain another group that will be added to the pyruvate, and this molecule in fact is called the coenzyme A. It is a small molecule that takes part in several reactions in the cell, and together with the remaining part of the pyruvate, it will form the acetyl-CoA or acetyl coenzyme A, and we shorten it into acetyl-CoA. During this process, there will be an exchange of electrons and protons, and this will give rise to the production of an NADH molecule, which is again one of the electron carriers that will later be able to provide energy by the means of oxidative phosphorylation, a process that we will review in the second part of this chapter. So now that we have our acetyl-CoA, it will be able to enter into the citric acid cycle. We say that this is a cycle because the molecules entering in it will end up with end products that will be able to re-enter in the cycle just with the addition of entering acetyl-CoA. Acetyl-CoA itself will combine with the final molecule of the cycle, which is called oxaloacetate, and in the first reaction will lose the coenzyme A and form the citrate. So if you understand this well, you see that the coenzyme A just served as a shuttle to bring the pyruvate lose one CO2 first, but it brought the remaining part of the pyruvate and incorporated it with the oxaloacetate into a molecule called citrate. So this citrate molecule will first lose one water molecule and be converted into cis-aconitate. This is a reaction that doesn't consume and doesn't release energy. Now that we have the cis-aconitate, it will be converted into the alpha-ketoglutarate, and this reaction will produce one electron carrier. It will also release one CO2 molecule, but the electron carrier will be used later by oxidative phosphorylation. The alpha-ketoglutarate will be converted into succinyl-CoA. So again, we have the coenzyme A that will serve as a shuttle. So the succinyl-CoA formation is accompanied by the release of a CO2 molecule and the release of an NADH molecule. Again, an electron carrier. Then the succinyl-CoA will be converted into succinate. So it loses the acetyl-CoA part, the coenzyme A part, but it will give rise to the succinate. This reaction releases a lot of energy that is taken for the formation of a GTP molecule from GDP, and this molecule will be further converted into ATP. So it is the first ATP formation from this cycle. The succinate now will be converted into fumarate. The fumarate formation is accompanied by the production of another electron carrier that we have never seen before, the FADH2. It has the same function as the NADH to carry high energy electrons. The fumarate will be converted into malate, and finally the malate will be converted into oxaloacetate, again with the formation of a NADH molecule. So the net yield of a citric acid cycle turn is 1 ATP molecule and 5 NADH. So here you have only 4 on this slide, but the 5th one is on the previous slide. It comes from the formation of the acetyl-CoA itself. And each glucose molecule will provide 2 acetyl-CoA molecules, so we'll make 2 turns in the citric acid cycle. Now, the citric acid cycle is a common cycle that can be used by a lot of entering molecules, and acetyl-CoA basically can come not only from the glycolytic pathway. In fact, when it comes from the glycolysis, it comes by the way of the pyruvate that is transported into the mitochondrion and converted into acetyl-CoA. When it comes from fatty acids, in fact, the fatty acid is already in the mitochondrion, and each carbon molecule removed from the fatty acid tail will produce 1 acetyl-CoA. And when it comes from the amino acids, it can come from the pyruvate that is transported into the mitochondrion, or it can come from a direct entry by the means of intermediates like amino acids that are partially metabolized and will enter in at various steps of the citric acid cycle. We will see that in the next part of this chapter. So overall, again, the ATP yield, direct yield of ATP from the citric acid cycle is only 2 ATP, and most of the energy that will be gained from the citric acid cycle will be by the means of the electron carriers, the process that we will see in the next slideshow. So overall, if you look at the ATP production, direct ATP production from the glucose molecule, you have 2 ATP molecules coming from the glycolysis, 0 ATP molecules coming from the acetyl-CoA, 2 ATP coming from the citric acid cycle, and overall it will make a 4 ATP yield for each glucose molecule. If you just look at the direct formation of ATP, it is called the substrate level phosphorylation, so the direct addition of a phosphate on the ADP molecule, okay, forming the ATP. Most of the energy, as we said, will come from the high energy electron carriers that will enter into the oxidative phosphorylation process that is covered in the next slideshow.